Hello, you all. My name is Nicole Mullen, and I'm an undergraduate researcher in the Gilman Lab at the University of Oregon in the Institute of Molecular Biology. I'm so excited to share with you today my research, which explores the role of the microbiota in the development of insulin-producing cells in Drosophila melanogaster, otherwise known as fruit flies. So before we get too far into the weeds, an important piece of background. All animals, humans included, are complex systems of interacting host and microbial cells. So I want you to think about your arm and imagine all of the microbes living there, interacting, competing with each other for space on your body. I also want you to think about your gut. That is also a huge area of microbial interaction. Most studies before focused on pathogenic bacteria or those little microbes that can make you sick. But we're in the midst of a really exciting paradigm shift where researchers are slowly realizing how impactful the microbiome can be for human health and disease. Chronic diseases, including diabetes, cancer, and significantly for my research, metabolic functions and systems are especially implicated in this. If you look at Figure 1, this illustrates previous research in the Gilman lab showing that the resident bacteria are required for the normal number of insulin-producing cells in zebrafish. And this was also replicated in mice. A secreted bacterial protein called beta cell expansion factor A, which is seen in Figure 2, was found to be sufficient for the expansion of this cell type. BefA is a bacterial protein secreted from a commensal bacteria in the larval zebrafish pancreas. In flies, the equivalent insulin-producing cells, or IPCs, are located in the brain, with 7 IPCs located in each lobe, as seen in Figure 3. My research worked to see if the relationship between the microbiota and the development of insulin-producing cells is conserved in fruit flies, as we saw it was in zebrafish and in mice. Doing this has the potential to show a powerful relationship between metabolism and microbiota observed across multiple model organisms, and will also contribute to important research showing that the microbiota is essential for the normal development of insulin signaling pathways. Because diseases like type 1 and type 2 diabetes both involve a loss of beta cell mass and function, learning more about what influences the development of the insulin-producing cells in any kind of model or organism could be critical for directing treatment in diabetes and human diseases. So I have 4 aims in this research. My first aim was to investigate if the microbiota is required for the normal numbers of insulin-producing cells in flies. Once I established this, for my second aim, I tested whether feeding the fly larvae the BefA protein could rescue the diminished IPC numbers found in germ-free flies, as it was also found in zebrafish. My third aim, test whether transgenically delivered BefA or BefA delivered through the actual genome of the fly also had a rescue effect. And finally, my fourth aim, test whether the transgenic expression of TSL, which is a naturally occurring pore-forming protein in Drosophila, also had a similar rescue effect. The reason I'm testing TSL is because it's hypothesized that BefA functions by perturbing membranes. And since I know that TSL is already pore-forming, I can compare the effects of both BefA and TSL in the fly genome and see how that influences the development of IPCs. For my first aims, I counted the number of insulin-producing cells in each brain lobe using fluorescent microscopy. This isn't as simple as just looking inside of a brain and counting all of the cells. In order to count these insulin-producing cells, I dissected, stained, and imaged third instar larvae that were expressing green fluorescent protein, or GFP, in connection with DILP3. And DILP3 is a Drosophila insulin-like peptide gene. This made it so that when DILP3 is being expressed, the insulin-producing cells glow green, as seen in this figure 7 here. This allowed me to count and compare the number of IPCs in germ-free flies, where all of the microbes have been removed, and compare this to the number of IPCs in conventional flies, where all of the microbiota is left intact. For the feeding assay shown in figure 5, all flies were fed a liquid diet of sucrose and bromophenol blue dye. Because larvae are translucent, it was really incredible to put the dye in there because it served as a check to ensure that the larvae actually ate the food. The experimental group of germ-free flies was fed the same liquid fly food with added BefA. And for the transgenic expression assays of BefA and of TSL, we used the specific GAL4-UAS system to express these genes in the tissue type that we wanted. The GAL4-UAS system is an incredible system that allows a scientist to express a gene in question, whether this was BefA or TSL, in a specific tissue type. So in this case, I expressed it in the eye and the IPC cells. I then compared the IPC counts between germ-free flies, conventional flies, and my experimental group with the BefA or TSL transgene insertion. So you all want to know the results, right? Well, of course, in addressing AIM-1, brain imaging and IPC counting demonstrated that germ-free flies have fewer insulin-producing cells per lobe than flies that were raised conventionally. If you look at that graph here, we have 2 representative images of conventional and germ-free brain lobes, and the germ-free has significantly fewer IPCs per lobe than conventional. Conventional rested at about 7 IPCs per lobe, while the germ-free were closer to 5 to 7. If we look at you can also look at that graph there. We have our experimental groups on the x-axis, my conventional, and then the germ-free. And of course, on the y-axis, we have the number of IPCs per lobe. And it's important to note that for this quantification, I looked at IPCs in 156 conventional brain lobes and 106 germ-free. So once I established this profound difference between conventional and germ-free, I went on to my second aims. In addressing AIM-2, the BefA feeding assay showed that feeding the larvae the BefA protein can partially rescue the germ-free effects, of the larvae. So this just means that feeding the BefA protein brought those flies closer to conventional levels. When I was looking at, and you can see those 3 groups on the x-axis. We have conventional or CV, germ-free, and then the germ-free plus BefA. And you can see that the difference between the germ-free and the germ-free plus BefA was statistically significant. When I went on to my third aim to see whether the transgenic expression of BefA also led to an increase, I found that yes, between the conventional and the germ-free that also expressed, between the conventional germ-free and the germ-free plus BefA that was expressed transgenically, all of that on the x-axis, you all are pros at this now, and the IPCs per lobe on the y-axis, I saw a profound difference between the germ-free and the germ-free plus BefA. And this suggests that BefA functions similarly in flies than it does to zebrafish and mice. And finally, when I looked at my last aim, which was testing the transgenic expression of TSL, that pore-forming protein found naturally in fruit flies, I found that this one actually had the most profound rescue effect for the germ-free treatment. Again, we have that final graph in figure 9. We have conventional, germ-free, and then the germ-free plus the transgenically expressed TSL. And you can see that the germ-free and the germ-free with the transgenic TSL had the most profound difference in IPC numbers. And this suggests that a pore-forming mechanism may be involved in the normal development of insulin-producing cells. So what does all of this mean? Well, first, my research shows that the microbiota is necessary for normal IPC numbers in fruit flies, as a germ-free treatment led to a decreased level of IPCs. I also learned that BefA protein alone, when delivered through a feeding assay, is sufficient to rescue some of those diminished IPC levels. Next, when I added BefA delivered transgenically, it also showed a rescue of those IPCs in germ-free larvae. And finally, when I tested transgenically expressed TSL, I found that this too led to an increase in IPC numbers for germ-free flies, suggesting that a pore-forming mechanism may be involved in the development of insulin-producing cells. Future experiments will focus on the mechanisms of BefA. We've already looked at some things where we add BefA to a vesicle and have seen that it is having a membrane perturbing ability. And we're also trying to find out where exactly BefA fits into the insulin signaling pathway. Preliminary results from a developmental assay suggest that germ-free treatment also leads to a slower development of the flies as a whole. And this is profound because it would connect the development of IPCs, or those cells that we see in the brain, to an actual change in the time period for the growth and development. This is exciting because beta cell loss of beta cell mass and function is implicated in metabolic dysfunction and diseases like type 1 and type 2 diabetes. So what have you all learned from all of this? Well, we know that the microbiota has a fundamental effect on the metabolic pathway, fundamentally affects cell growth and development, and because of the location of these IPC cells in the brain of the fruit fly, could potentially affect the brain and have implications for microbiota's effect on the way that we think. None of this research would be possible without all of the generous support from the funders. I would like to thank the University of Oregon SPUR program, the National Science Foundation, the National Institutes of Health, and of course, all of the members of the Gilliman Lab for their incredible support of this research. Thank you so much.